Welcome to the Dr. Shawn Tierney Podcast. My name is Shawn Tierney, and I am a neuromusculoskeletal sonologist. I treat athletes and high performers. My goal with this podcast is to help you make better decisions regarding your health, athleticism, and all aspects of life. Here, I'm both a student and a teacher. Thank you for tuning in. Hello, this is Dr. Shawn Tierney. Welcome to the Dr. Shawn Tierney Podcast. Today, I'm super excited to have a friend of mine, very respected scientist in my world, Adam Koster. Adam Koster is an authority in my experience in regenerative medicine, stem cells, A2M, exosomes, extracellular particles, and just a guy that I learned a lot from. And the thing that makes him different than other scientists that I've worked with, and there's a lot of great scientists that I've worked with, is that his observations kind of go along with what we observe clinically more than other scientists, and so he kind of puts the picture together well. He's a great person, a great friend, and Adam, tell us something about how did you get to this place? Because I know it's been a journey for all of us to get here.
Yeah, of course. Great to meet everyone. My journey began at the bench originally. I worked on different versions of MSCs or mesenchymal stem cells and iPSCs, induced pluripotent stem cells, all the way back in 2008, 2009. During my education, got a bachelor's degree in neuroscience, was always fascinated with the brain and nervous system and spinal cord. And, after spending a couple years doing some animal work, really, on Parkinson's disease, also Huntington's disease, and at the Brain Institute at Central Michigan University, I was fortunate just to do this hands-on. So every layer of cell culture, to producing the cells, to maintaining the cells, to understanding, how to introduce them into animals, track and study those animals. I was very, very fortunate to learn from Dr. Gary Dunbar, who runs the Brain Center up in Mount Pleasant, and then had a mentor named Kyle Fink, who's since gone on to the UC system with Jennifer Doudna and, discovered CRISPR, and they're doing amazing things with the cell therapy program at UC Sacramento and all the different subsystems there. So I eventually transitioned into the commercial world after kind of discovering working with mice and rats was not exactly what I wanted to do. I'm more of a people person. And like you said, Shawn, my experience has been interesting because it's, for the most part, always overlapped with clinicians. So early on, and when I got involved in regenerative medicine, which was going all the way back to 2010, this was just when some of these devices in the OR were getting their start with centrifuges, where we're taking blood, we're taking fat, we're taking bone marrow from the patient, removing them with sterile systems and medical devices, processing them in some way, shape, or form, concentrating, removing, adjusting, and then working with clinicians like yourself to put them back in. And over time, I was fortunate to go to many, many offices, hundreds and thousands of clinicians' offices, not just in the U.S., but in Germany, in Australia, in Asia, in South America, Central America. And, just like any career path, I think over time you learn there's a lot of people that are pretty good, and then there's about 5% that are just rock stars. And I would gravitate over time to these clinician rock stars who really, their expertise with these different products, and then over time stem cells, and then over time exosomes, it came about because of experience. So they all started in the traditional system, all insurance-based, all hospital-based, whatever that may be, in surgery centers. And then eventually, just found a way to slowly either transition to just cash. Some of them just still do a blend of insurance and cash. But they do what works. And they've figured out what works over time because they've tried a lot of things. And I don't say that haphazardly. You can think of these as really clinician scientists, right? So as these different blood-derived, fat-derived, bone marrow-derived, and then off-the-shelf or allogeneic options from birth tissue have found their genesis, over the last decade and a half, a lot of this trial and error process has been done in tandem, in collaboration with people like myself, making and innovating these products along directly with the clinicians like yourself who, a lot of reasons why I gravitated towards you, right, is your portion of getting things in the right place, but not only understanding the idiopathy of what's really going on and not just maybe injecting a bolus of something, not blindly,
Hoping for the
but
best. Yeah, the magic.
right.
Nothing is really magic, but we have some really good tools.
Yeah, and I think that over time, when you're actually doing it, and obviously I'm not a PhD, I'm not a master's, but I have intimately studied extracellular vesicles and MSCs and stem cells for well over 15 years, and so most of my education outside of my time at the bench and my degree in neuroscience has been hands-on, right? And that not only spans from, I've been in many ORs helping with the processing of these products, the blood products, the fat products, the bone marrow, intimately understanding what matters to me the most, which I found over time didn't always matter the most, which is when we're putting this in, even if it's coming from our own body or if it's coming from off the shelf, what's in it? What's actually in this, right? How can we understand what's in this? And
yeah, you're asking deeper,
I was always just so motivated to drive towards working with clinicians who rarely had those answers, right? Because clinicians are busy, right? They're running their practices. They don't even know the hardware used to be able to test what's in something, right? And you and I've talked about that extensively. And that's what's so cool about your background is you were at one point an analytical chemist using, HPLC and LCMS, which we can talk, to the audience a bit more about. But
yeah, well, I had my brief experience, nothing like yours, just working with environmental chemistry, HPLC, mass spec, water soil analysis for in California. So I understand a lot of the equipment. I am not at the level of expertise that you are. I think I appreciate where you're at, and I appreciate not only have you been through this, as far as the science goes, I think you are more in touch because your experiences as working with clinicians has put you more in touch than the average scientist out there. And like, hey, what actually works? And I always go back to my goal as a teacher, because I teach ultrasound guided diagnosis and procedures, is to get people to see the truth, right? And that's what we're all trying to do, is see the truth. And there's always an ego or a paradigm that can get in the way. So I think as we come into a situation, we want to look and say, hey, how do I get through my ego, my little point of view, and see other points of view and other paradigms, so I can actually see the truth. And it's when we're dealing with people, that's important because we want to solve a person's problem. And if we're not willing to see the truth for ourselves, we're not able to critically look at ourselves, critically look at the scientists and the papers and the information that we read, and say, hey, sometimes this just doesn't work. This is not the answer. We need to go back to the drawing board. I think there's a humbleness that's required for that. I think that Adam has that, and I think that a lot of the scientists that he works with have that. There's a lot of outstanding people in these fields. And what I see in you is not only do you have that understanding of what clinicians are looking for, but you also have the knowledge of how to build your own company. You have your own company, and you also have experience with dealing with the regulatory industry and kind of guiding something where we can actually make a change that helps humanity. It's a complicated world, but we have, a piece of this picture that has a lot of answers, and
you make some really good points there, right? Because regardless of whether we talk about nanoparticles or stem cells or the FDA or, how that relates to big pharma and all the implications there, I think at the end of the day, where I came from and the way I am, why I am, is mostly due to open-mindedness, right? And the willingness to fail a lot and admit that I don't have all the answers, but I am always voraciously seeking people like yourself who think the same way, but because of their experience and they've done things, right? They're not out here talking about a lot of things, but they're doing a lot of things. They're action-oriented people. Just from early on in my career, yourself, Dr. Michael Wolf, Dr. Sheldon Jordan, Dr. James Secret. I mean, I could go on and list about 100 people, right, who I knew very early on if I just gravitated toward their credibility, gravitated toward their skill set, gravitated toward their values and their ethics and their genuine unicity, how they treated patients, and how they navigated a difficult system at times as well, right? Without losing their self. And also, without becoming, I think, too blindered, right? I think oftentimes in regenerative medicine, this has been a bit of a crazy world, over the last 15 plus years. And people can be very territorial, right? I think having an open mind about what's possible and the potential of not just what can come out of our own bodies and go back in, but potentially what can come off the shelf and how it can augment some of those things or complement some of those things, I think is quintessential to the future of what we're trying to do. So I have been very fortunate over the last 4 years to build a biotechnology company, build a real team, producing real science, real results, real products. And I think, the best part about that is you start to just welcome this high level, I would call it the suffering. Like after so long, you are so used to dealing not just with problem solving, but doing really hard things all the time, that you just frankly, like after a while, become numb to the thought of a hard thing. Like you just wake up and something will blow up and you become very unfazed, right? And that in and of itself is something I'm actually grateful for. I think, I was thinking last week about getting, into an MBA program. But I've been trying for years, right? To do this. This is a goal I've had. And I was thinking about like telling, my family and telling some close people to me who helped me get there. And it's really not for me about the destination anymore. Like I'm excited to go do the thing, but I'm so much more excited about doing the thing and getting there right now that I understand really what that means. And that means so much more than me. That means my team, that I'm responsible for, and, you want to enjoy it. My greatest mentor in life, Dr. Frederick Naftalin, who is a former chairman of Yale, one of the top institutions in the world, and has come alongside of me at 80 some years old, who why me, because we do incredible things together. Right. And, he's taught me so much, and, some of those things, like nothing is wasted, right? There's so many days and there's so many things where you're just, man, back against the wall, things are going wrong. But if you're constantly looking at things like, what am I learning because of what I'm going through, right? If you're shifting your frame and remembering like, hey, we didn't do this to like hate this. We did this because we want to make people feel good. Right? Like, what is our end goal
solve
here?
Problems, right? Yeah,
we want to solve problems. We have to solve problems. That's what we have to do. But I don't wake up to do this every day just cause.
Yeah.
I wake up to make people feel good, right? And that's a big thing to me.
That's kind of what I call professionalism, because I think, there's this attitude where you get educated and you do something and you're doing it and you're excited about doing it. And in your world and in my world, that's not enough. I always say, when somebody comes in and they have and I see that they have a pain in an area or something wrong in an area, there may be 7 things wrong. Now, if I missed one of those 7, they don't get better. They're still in pain. Nobody's happy. Nobody wins. Nothing got completed, really. That's kind of what you're saying is here, is that, you're a professional. You're looking at all the different aspects of these issues, and you're, with a humble mind, you're looking at all the perspectives. You're not just judging by yourself. You're looking at opposing points of view, but then you're coming to this place where I know this is going to work because I covered all my bases. I covered all 7 things that could, or 7 or 100 or whatever they could go wrong. And so now I can move forward with confidence. And that's professionalism. That's consistency. That's, you want to be consistently successful and constantly willing to fail. It's kind of a hard place to be.
Yeah, and
that's
it's also like
yeah.
some of the things, that you do and that I get to do. These are not like, let's find the result next week, right? I think there's a beautiful aspect to what you do where you do get some of that immediate gratification with your patients where you get to see it on the table. But and I juxtapose that with myself and our team when we look at like just one regulatory submission, right? You look at that paperwork before it's not filled out and you just go, are we sure we want to do this? This is a very long-term thing. And yes, like you have the greatness of having that, more near-term gratification of helping your patient. But I think what most people wouldn't immediately realize, it took you years to learn those skills and develop that acumen and that expertise and gain that experience to be able to hydrodissect all the cranial nerves or hydrodissect. I mean, these are incredible things that you're doing, right? But I think thinking about that and having that long-term mentality, but also like knowing that you can only do so much each day. And so even if it's 1 brick a day, right? Even if one day, like you make a brick, but it falls apart. Well, you made a brick, right? Like you broke the brick, but tomorrow you can try to build the brick again, knowing how you broke the brick yesterday, right? So
you're going the long haul, but you're saying, this is stuff that is, changing people's lives. And it's, we're basically covering basis in healthcare that's not covered in other ways. So, you're probably like me. You don't want to do what everybody else is doing, and you want to solve the problems that other people are not solving, because solving the problems that are easy to solve suddenly become less interesting over time, right? So as a result, you end up being there. And I think that there's some innovative things that are coming up, and some egos that have had to overcome. Because I know that one of the things you're dealing with is it the excretions of the cells or the cells that make the difference, right? And how can we manipulate these things? How can we do we need to manipulate them, or is it just what our cells make? So this area of creativity to some extent is exploding right now in your field of cellular medicine, regenerative medicine. But it's also at the same time, almost has a heavy hand to push it down by regulators. And so the idea is, well, maybe sometimes we have to take this stuff and do it outside of the country. Mm-hmm. But if you're going to do something outside of the country, at least you want somebody who knows how to do it with the highest level of regulations. So if it's out of the country, it's super, super safe, as if it was, regulated in the United States. In a safe way, provided that some of the regulations, it's just not going to happen in the near future, that we can do some things. So you're offering the ability of people to go to other places, maybe in everything not in the United States, but go to other places and have something done safely. And I think that you and I have both seen, I don't want to talk where people have gone to other places and they didn't have somebody with the safety profiles, with the knowledge, and things haven't worked out so well.
Yeah.
So we have this way to be, I say, professional and not make a mistake and not take a risk of losing an important battle in somebody's health.
Yeah, look, I think that this world of regenerative medicine, and as I've walked through my journey, which went from the use of just off-the-shelf FDA-cleared medical devices at first to take out stuff from our own bodies like blood, bone marrow, fat, like we touched on, where that there's a pretty high safety profile. Those products have been tested and measured and consistent, and they're sterile. They're closed systems, so you can control the issues that may be encountered, and it's coming from the patient, right? So there's not a lot of issue of it being rejected or causing any sorts of, crazy problems that haven't been studied at nauseum. These are all important things to consider, but I think that really what we're going to discuss today, which is the use of stem cells, the use of exosomes, these nanoparticles that exist inside of all of us, it's really just kind of like the next step, right? I think it's already been happening for quite some time. And I think that regardless of the goalpost not existing right now with the FDA, regardless of it being we're really early adopters, probably even earlier than that to a certain extent. We have to really inform the audience of what the facts are, right? Which at the end of the day, the stuff that everyone has been using, like the blood, the bone marrow, fat, what's inside of those products? Cells, right? Platelets, adipose stem cells, bone marrow stem cells, cells release exosomes. So we're already doing these things, but we're doing them in a goalpost or a paradigm that exists, right? So the next step is to do it with what's, the natural progression of what's next, right?
Right.
Which is supercharging that. And
Right.
so and then to me, that becomes more specific, more tailored, more precise, more engineered, whatever that may mean. And we'll get into that. But I just wanted to draw that characterization of, when you were talking earlier about the importance of open-mindedness and genuinicity, well, those principles of simplicity are what drew me to exosomes in the first place.
Right.
Because I'm just a kid, it's so excited about biology, and I learned that our bodies already have this natural drug delivery mechanism baked into them already. We already have these nanoparticles, and they're already the highway of how our cells communicate, and this paracrine system, or this crosstalk of the cells talking to each other. You think of a cell, it is essentially a little bubble of fat, right? And you think of a cell as the size of a quarter. Well, the cell will release a particle or an exosome. It is 1/1000th the size of the cell. So the quarter's on the table. You can't see the exosome next to it, but the amount of exosome released in comparison to the cell, X factor, right? Like so many more are being released. So what's small, released in high enough of a bolus, can cause amazing things, right? So at the end of the day, a cell is a bubble of fat with a little bit more complicated machinery, right? A nucleus, organelles,
A lot
and
complicated, but yeah.
different behavior, right?
Yeah.
Sets the tone of environment and leads to organs and systems that build us, right? These are
Right.
living beings. We are living beings. So these are the layers that really fascinate me, right? When we get to kind of talk the basic sciences. And I think, helping everyone understand that simplicity is probably one of the most important parts for me, because as it gets more sophisticated, as we try and engineer how these cells behave and engineer the nanoparticles that they express and release, okay, yes, that's a bit more complicated.
Right.
And we can get into that, but I think understanding how to get into that and how to educate people on that, you need the baseline. You need to really understand what this is. And it exists inside of all of us, which is the
Right.
coolest part, right?
Yeah. So the paradigm shift that I think we're talking about here for everybody is, there's this idea that there are stem cells that are magical, that will cure things, and I think we're saying, well, maybe that's not true. Maybe it's what the cells produce that's making the difference, and how can we make the situation safer? We talked about like PRP. PRP is one of the first things we used in regenerative medicine. It's good. There's some things can go sideways with it, where you can have a flare, things get worse. So then we're like, okay, well, that was okay, but is there a way we can do something that's not going to be as risky, that's going to be safer, that's not going to put somebody in jeopardy, that we're not going to get worse before you get better? And then there's the next step, which we're going to use bone marrow. We're going to use fat. Okay, then we can use the secretions of cells. And then all of a sudden, wait a minute, and you knew this way before I did, that's probably where it's at. And whether it's you have all the secretions, like the soup of everything that makes you grow and heal, or just a specific one. So you want to kind of delineate out. Okay, we have these secretions that are very specific, or we have all the secretions, and we can put drugs in there. That's what the pharmaceutical companies want to do. Or we can say these cells do this. This is what we see happening. This is where the lab becomes a creator, versus are we going to make the lab the creative force, where you're not going to have billions of dollars to produce an individual product, or are we going to have, a pharmaceutical company make something that's very synthesized, where maybe it'll be 50 years before you can make a product that works as well as what the cell makes. So what direction would we go? And I think the FDA doesn't know what to do. But what we want is you to get better, right? What we want is results for you. What we want is to improve humanity. How do we do that? And I think, Adam, you're on one of the most direct paths that I've seen. Obviously, right now in the United States, you can deal with cosmetics.
Yeah.
So I want to talk a little about that.
All these areas are just, this is the cutting edge, right? So I think it's so important for the audience, for us just to take a couple steps back and kind of just help them understand the basement understanding of a stem cell even, right? Because before we dive into these exosomes that the stem cells produce and why they're beneficial and what's inside of them and how they function and why they're potentially safer as opposed to using a cell. Arnie Kaplan, the original, creator of the term mesenchymal stem cell, progenitor cell, parasite.
I like it when you say it the American way. People say mesenchymal, that's the British way, American way.
Yeah,
anyway, whatever.
This was the word, man. I remember when I was first getting in 07, 08, 09, like MSc. It was like, whoa, we're going to talk about MSCs. Everyone wanted to talk about stem cells, right? And what's interesting is over time, and the late, great Dr. Kaplan, who unfortunately we recently lost, he passed on. At a certain point, he changed the name from mesenchymal stem cell to medicinal signaling cell. And so it's really, really important to understand, okay, what is actually a stem cell? And we threw out other words, parasite, we threw out progenitor, and you're going to hear some of these words. But what do these words really mean, right? And I think the understanding of these words is so crucial to re-educate the audience in understanding this paradigm shift around the word stem cell. I think the Bush administration and the issues with fetal cells and so much confusion, was caused with the general public. But in actuality, fetal cells are rarely, if ever, used, in any kind of treatment paradigm, mostly due to, some preliminary research showing that they could form teratomas or tumors, mostly related to aspects of their DNA in the fetal state. We do not work with fetal cells. We do not work with aborted fetuses. The majority of birth tissue products that are not, coming from your own body and being processed and going back in are used. They should be used from what are called AATB accredited tissue banks. It's a process you have to go through, get certified by the FDA. And if you've ever had a baby, you can bank the tissue. You can bank the umbilical cord for later use, right? And some of the only cell therapies that are approved are related to, stem cell related disorders, right? Graft versus host disease, for instance, where in leukemia, essentially the person's blood is so ridden with, cell-free DNA and oncogenic DNA and all sorts of really just toxic problems. You can essentially wipe out that person's blood and revitalize them with their bone marrow, right? And so when we talk about disease in the human body, it's so important to talk about disease in the context of a specific organ, right? We're system. We're really a system. We're an intricate. There's cells that form tissues, and tissues form organs, and organs form systems. It's this incredible
Yeah.
symphony of all these things like working in tandem. You start, studying the pancreas and how it interacts with the liver, and how it interacts with the gallbladder and the surrounding organs, and how the hypothalamus controls the release of all these hormones and the time. This is
Overwhelming.
why
Yeah.
I
It's
got
a lot of
obsessed
good
in the
stuff.
first
Yeah.
place,
Yeah.
right? So I think looking back at organ-specific disease perfectly ties in with stem cell-specific cells for those organs, right? So, I think as the therapies go to the next level, we're going to see what are called progenitor cell-specific or parasite-specific cells for that organ, which are essentially the resident stem cells for each organ. If you go to a stem cell in your kidney, and then you go to a stem cell in your liver, they're going to look very different, right? They're slightly differentiated towards the environment of that organ. So it's going to have a little bit of a fingerprint more towards that region, that environment, because it's been exposed to different things. It's, been going down its lineage to a little bit degree, but it's still at its origin, right? And that's the way that we need to think about it. Like, this is like the cell at its most naive or its most native form, right?
The purest form of that cell in that organ. But even then, it seems like, using that pure cell versus using a more earlier stage, like a mesenchymal cell, maybe that doesn't make as much of a difference as using what that cell produces, right? So then it comes back to, because that's the argument that I'm not sure if you heard from other physicians, like, hey, we can delineate these cells, but delineate these cells, and we give that to, and we using those cells doesn't really work better than the mesenchymal cells. But when it comes to the cellular excretions, now we have something that's really, powerful, different and more powerful.
Without a doubt. And I think when when the literature is now showing, as they're labeling and tracking and monitoring cell-based approaches, right? And I'm talking MSC-based approaches. Oftentimes these cells are persisting in the region, or they're given IV, or they're given IM, or, they're injected into a joint. The cells will persist in the area for a number of days, and they will traffic out. Many will die, many will not survive, and almost all of them will be dead or have some form of senescent and then turnover within, a couple of weeks up to maybe a month at most, right? And so what's happening there? The cells are not engrafting. They're not turning into cartilage. They're not turning into bone. They're not turning into the resident cell type in the tissue. So what's happening is they are releasing signals, medicinal signaling cell, right?
And that's where it comes down to. That's what kind of drew my interest towards, Adam's field here, because what I saw was, even if you put the purest, maybe never frozen, freshly produced cells from yourself into yourself, they don't survive, many times. And especially if there's not if you put in, a cell into a joint, well, there's no blood supply in the joint, the cells die. That causes a problem, inflammation, pain, and sometimes things get worse, even though you had some great, what we thought was great stem cells.
Well, again, they're living, remember.
Yeah.
So when I think of a cell, I think unpredictability.
Yeah, exactly.
Over 40% of FDA cell therapy clinical trials have been placed on clinical hold. Why? Because cells are highly unpredictable, right? Cells are their own single unit factories of activity, right? And the cells around them influence each other's behavior. The tissue niche they're in influences their behavior. Sound influences their behavior. Like, we did some work in cell culture, and having my colleague Ahmed, he has grown cells in flask bioreactors or at scale in various institutions, Yale, NYU, Vanderbilt, and others, for 40 plus years at the bench. Ahmed knows cells grows better in the morning. They double better in the morning. He's observed music being on, and cells behave better. So these are living beings, right? And I think it's important to remember that. And they behave based off their environment. the process is where I've learned the most, trying to produce specific exosomes, or really the hardest parts, which is producing consistent exosomes at scale, all relate back to the behavior, the conditions, the environment that we put that cell in, right? If we find the best, most special cell we can, wherever that comes from, we have to have the right media. We have to have the right size, shape, culture flask or bioreactor system to cause a stir tank effect. Like, all of those little layers and nuances matter so much, because otherwise you will not be able to have a consistent process that literally leads to a consistent product at scale. And I think that's the field's biggest issue right now, especially with companies that commercialize products into the regenerative market very quickly. These products no doubt have exosomes in them. The stealth products no doubt have cells in them. But, I myself have tested over a dozen products in the market. They're all inconsistent. None of these products are consistent. Right?
And that's the issue that I've observed, in clinics that, I go to many clinics and help guide procedures, diagnose conditions. What I've noticed is that, hey, these things become unpredictable. You can have a reaction to your own cells. You can have a reaction to somebody else's cells. The cells can die. This is just, you lose that predictability. And it's like when I say, hey, I think being a professional, being really good at what you do, people want to see you. You don't really want to have anything go sideways. Don't want anything go the wrong way. So when you're using cells, things go the wrong way. Too much for me to be comfortable with being a part of it. In many cases, not in every case, but in many cases, things can just go sideways. And even if it's only a small percentage, if you see enough cases, you don't want anybody to have a bad experience. You don't want anything to ever go sideways. So then how do we make things never go sideways?
Well, this is the FDA's job, right, to a certain extent. But
exactly.
I think this has also been one of the biggest gaps with clinicians, right? And I wouldn't say it's their fault. I definitely wouldn't. I would say that once you start to, again, this is the hard part. Unless you go through the process and understand donor tracing, traceability, batch records, and donor variability, and all the layers and aspects of growing a cell therapy product, you won't get it, right? So, you can look a lot of these things up on the FDA's website. I think a lot of clinicians don't realize that, but they don't have time to do that, right? So that's been a big skill set of mine, is a lot of times I'm just directing clinicians to resources that are already there. It's just I'm turnkey making it available. Here's the information. I'm just presenting the information, right? So, I think that as the FDA catches up to a certain extent, right, if you look at many, many companies that are making birth tissue products, which, by the way, birth tissue derived allografts and sheets and matrix and flowables and cell products have been widely used and approved and covered by insurance for ophthalmologic use of the eye, for wound care, for spinal and orthopedic and surgical procedures for decades. This is not something new, right? This has been done for years and years and years. And plastic surgeons rely on these grafts for burns and wounds and, accelerating healing. So I think there's also just a drastic mischaracterization around, can these products be safe, right? They can be. And they have to be tested appropriately. They have to be safe. And there's ways to do that. But I think that the field, to a certain extent, has just been marred and a little volatile over the last decade, I would say, because there have been so many companies that dive in and don't understand GMP, or good manufacturing process, don't understand donor traceability, don't understand batch records. Like the integral part of making a human-derived product is the quality management system. It is by far the most important part. And I think, unfortunately, a lot of companies, they start later with the QMS system, right? If the FDA shows up on your doorstep, which all of these facilities have to be registered with the FDA, and at some point they will, there's a very specific protocol. There's a very specific process. Show us this information from your QMS system. And if you look at the 483s and the warning letters, which again are publicly available on the FDA's website, we've all seen many of these warning letters coming out over the last, and not just to labs, to clinicians for marketing and all these different reasons, right? So time and time again, if you look at those warning letters, you can look them all up. I could list 50 companies right now that have come and gone because of these warning letters and problems. It's related to sterile techniques, sterile procedures, lack of protocols for gowning and sterility and safety testing and aseptic technique and donor traceability and all the things that I just keep mentioning. It's the same dings over and over and over, which is vital, right? Because this is what leads to contamination. We saw the outbreaks in Nebraska. We saw, the unfortunate cases that happened down in Florida where the patient went blind. So things can happen, right? If these products are not third-party tested and even tested third-party for consistency, as a part of our regulatory applications, cells, because they're so unpredictable, and because the environment you put them in can cause them to behave unpredictably, what they're secreting can be widely variable, right? And so we have to control that variability. And we don't just have to control the variability from the aspect of a consistent product, also from a safe product, which means every step of the way, there has to be validations. Can you show what's in it in a measurable way at this point? Can you show that it's safe in a measurable way at each step of the process, right? And so it's important to understand that process. We're growing cells in some vessel that's either a flask, a roller bottle, or a bioreactor where you're growing liters of cells at a time. And then we're using sophisticated filtration technology because once we grow the cells bathe in what's called media. And once the cells are in with the media, they condition the media. The cells release all these wonderful factors. We call them trophic factors or exosomes or growth factors or cytokines or all these wonderful words, right? They condition the media with this soup of this symphony of glory, right? That can have so many beneficial effects: pro-growth, anti-inflammation, modulating the immune system. I mean, the list goes on and on. Antimicrobial. And so after we take that conditioned media, and then there's various filtration steps and processes. One of those typically, if we're working with exosomes, and will involve an anion exchange or a type of enzymatic reaction to remove the DNA. We do not want foreign DNA in these products. The majority of exosome products on the market, they do not do this. So they almost all have foreign DNA in the products.
If you have DNA, then you can have immune reaction to that, and that's one of the issues. And what are the other issues with DNA?
Yeah, look, so it's not just cell-free DNA that exists in the media. It's also chromatin. And so chromatin can be very inflammatory, and it's very sticky, right? So these are deep scientific questions, and frankly, they're ones that I haven't studied enough to have the real answers from. And this is really what I do, right? Is I work with the experts when there's questions that I don't have the answers to investigate them further, right? And so once we do this filtration, potential anion exchange or enzyme treatment to remove the DNA, then there's additional purification steps. There's options like size exclusion chromatography, which uses HPLC, and very sophisticated capital and hardware that in the end gives us a highly, highly pure product. And now this kind of transitions into what we were talking about earlier, where maybe the most pure product on the planet isn't actually what's most effective, right? And so,
and we're not talking. When we say pure, we're not saying sterile. We're not talking about sterility. We're saying, is there other growth factors in there, so that all of the growing factors, or do we want certain growth factors?
Yeah,
I think that's
and
what
this,
you're saying. Yes.
And this, I think, is where the field is running into issues right now, is because as a biotechnology company, you cannot move to the next step without funding. You cannot move to the next step and obtain funding unless the venture capitalists or the banks or the financial syndicates have given the blessing that this is an emerging technology, it has promise, right? And so the venture guys have diligence people where they send them the data, they send them the science. And if our experience has been, time and time again, is this really a drug? Why is that? Is because this is a naturally occurring cocktail that we're collecting and purifying and extracting from cells that are potentially either already in our bodies or extracted from an ideal cell source, right? So this is a bit of the juxtaposition we're dealing with, which is a moral dilemma almost of we have to take something that's very natural and we have to figure out how to create intellectual property around it, which usually means making it more synthetic so that we can protect it, so that we can get funding for it, so that we can move it to the next step and test it in here. So it's a very strange.
Yeah. So it doesn't really fit the current paradigm of a drug, right? So we kind of broke the paradigm of we're going to have this chemical that does that, and we can patent this, and so what's going to happen with that? I think it's going to. I hope that when it ends up happening is we have, innovative people who are working safely to create these products that are essentially natural products.
They are and they aren't, right? I think they are very
native.
natural, but I think as the approvals come and the money comes, there's going to be Gen 2 and Gen 3 and Gen 4, which will be ratcheted up versions of something natural, right?
Yeah.
So what does that really mean? That means that at the end of the day, it's going to still look a lot like something natural, and to me, like the best cocktail is the dirty water, right? Like we take the ideal cell, we grow it, we produce the exosomes, we condition the media, we do some filtration and achievement of no contamination and safety profile and a solid understanding using LC-MS and nanoparticle analysis and PCR and these basic science techniques. And we what do we introduce? Almost 1,000 different kinds of protein. It's not one protein. This is nearly 1,000 growth factors. This is almost 500 sequences of nucleic acids, all kinds of RNAs.
Yeah, so you have this very complex solution, and which probably is maybe the most effective way to get tissue to change, tissue to become more healthy. But in the current pharmaceutical industry, if we were going to isolate each one of those strands of DNA and each one of those proteins, you're probably looking at 50 years before we can maybe be as smart as that cell is, right?
100%.
And that's so. How do we heal people today? How do we help people get through their problems today, using what a healthy cell would do? How do we enhance that? So I think that's the position we're in, and that's the position that I think the world is trying to contemplate. Because we have this investing system, like you said, all this stuff is invested. Is it just, hey, there's a good cell here, it produces really good exosomes, the stuff that it makes is pretty good, tends to heal these things. This is the cell that's a good progenitor cell from the Michael Jordan of healing livers, and this guy, his liver heals like a champ, and we get his progenitor cells, and we make those exosomes, and they're about as good as they can be. And the exosomes are not matched to an individual person, so we can make them very, very sterile and clean and safe. Is that what we want to do, or do we want to take your liver cells and make exosomes from them? That's kind of like where we're at, But I think it comes down to regulation. Kind of medical freedom issues as well. I think there's a talk of, are we going to have medical freedom where you can say, where you as an individual person can say, I want to do something that's not approved by the FDA, but I'm going to know that it's not approved by the FDA? Or right now the FDA says, well, it doesn't matter what you want to do, you can't do it because we don't approve it. So where is that medical freedom going to come in?
Mm-hmm.
I hope that it happens that you can have people innovative people like yourself, innovative scientists and innovative technologists that can push through this technology to the patient who needs it now, rather than, it's going to be working great 50 years later after you're dead, but it didn't really help you now. You're alive today. You want to be better today. You want to be through your problem today. So, it's like you said, it's a moral dilemma, but I think it's a real, a real dilemma. And I think that one of the biggest challenges to the whole system maybe was, I don't know if you feel this way, but I always thought was, there's some articles that came out out of Israel, Tel Aviv, during COVID, and said, hey, we have exosomes, these exosomes that we have, these mesenchymal exosomes that we have, they work great for COVID. Actually, we give them to some people that have COVID. Nobody dies. And if we make them for everybody, we can make them fairly inexpensive, and we can just end, and it's like in the paper, it's like, we can pretty much end this pandemic right now. And then I'm like, this is great.
Then it disappeared.
Make everything so cheap. Yeah, but I look for it, like, where's the next paper? It didn't come out. I'm like, this is exciting, and it's gone. I'm not sure, what happened, but I think it's a moral dilemma for the world. Obviously, everybody wants to have their piece of the pie, and how does that get distributed if you can just cure everything?
Yeah, look, I think you make some great points there, but I think the most important thing to take away from that is we're not going to jump down some conspiracy theory rabbit hole, but what we are going to do is talk about the regulatory bodies, right? Because we don't see them as the big bad wolf, right? Like the regulatory body, the FDA specifically, the big bad wolf. I like to call them that, but that's not what they are. No. My company is going to meet with them in a few months, right? Like they have doors. They have ways, they have means, they have mechanisms for early stage innovation to go and meet and ask questions. They have forms for this. They have processes for this. So, we're pursuing what's called an interact filing. And this is like a pre-IND meeting, right? Where we can put together our summary of all the preclinical safety studies that are out there and that we performed, our summary of all the clinical safety studies in humans, which there have been many with exosomes now, our manufacturing development plan, how are we actually going to make this safely at scale, and then our clinical development plan, where, people like yourself and their experience during that discretionary period where the FDA, said, okay, get the house in order, but this is a period where people were able to try some things, right? And they did. And so that dosing information and all those patients that were treated during those periods of time, the dosing information and the products that are treated in animals, there's a science to drug development, right? And a lot of that, the FDA wants to see animal studies showing where does it go? Where does it go and what does it do and is it toxic, right? And this is called PK/PD to a certain extent, pharmacokinetic and pharmacodynamic. What does that mean? Pharmaco, so a drug, right? Kinetic, the physics of where's it going, what's it doing, right? We all know kinetic energy and the concepts of physics. And pharmacodynamic, right? Drug. What's happening? What are the dynamics? What organs is it affecting? Where is the drug going? Is it toxic? Has it any ancillary effects to any other organs in the surrounding area, right? So, instead of saying like, well, we're too early and we don't know what to do and we don't know how we're going to get these approved, there's companies already doing this, right? There's a path that does exist. And so, irregardless of if we're going to create a genetically engineered transfected master cell bank to overexpress specific RNAs or overexpress specific proteins. So in the end, we still have that symphony. We still have the drums and the trumpets and the bass and the strings, and we still have that 1,000+ proteins. We still have hundreds of nucleic acids, but we genetically engineer the starting cells to overexpress specific instruments. So we're going to have a little bit more of one or two of them based on some positive results we saw in some animals.
A little more bass.
Yeah, I gotcha. little rigagoo-goo, So those are the things that we get excited about. But what's beautiful about the FDA is we can do that in a stepwise approach, right? Because genetic engineering is very expensive, right? Creating master cell banks, even more expensive. So, you look at genetic transfection methodologies, you go work with Lonza, you go work with Sartorius, some of these big other companies. You're not doing this for hundreds of thousands. You're doing this for hundreds of millions. This is how much it costs. This is not Subway. This is not a sandwich shop. It's a biotech, right? So, again, you cannot move forward and do the sophisticated things you want to do if you don't kind of do the basics that lay the foundation first. And so, That's what we plan to do. We plan to take a more unmodified version of our product to FDA, show that we can produce it consistently at scale, show that it's been tested safely in animals, show that we know where it's going because we've labeled it, using radio labeling and tracking, show that like every step of the process we can monitor that it's safe, that we have all these measures showing how we make it and what's in it. And that it's consistent, most of all, right? Which I think has been one of the biggest issues in the field. This is the path forward, right? And even though we open the document that the FDA provides us and it's 100 pages and we have to have the right consultant and our team is like overwhelmed and it's a lot, right? We get to bring experts like yourself who have so much experience with actually doing these things, right? So this is a really beautiful thing that we get to be a part of as a team. And as we, build the baby steps to get there, and whether it's producing, over the coming weeks, we're doing some batch runs to make exosomes in a bioreactor where we're going to be making a little bit more than we're used to making, right? Just to ensure that the consistency is still there, nail our process. And what's really nice is when we go to produce a 2-liter batch from a bioreactor, we can take that same protocol, that same process development that we spent months on, and just ratchet it up for a very similar piece of equipment. Same protocol, little bit more material, right? So this is our strategy, and based off some feedback from experts, this is our lead program. And it's not just my interest, and going back all the way to 2008 when I became a little kid obsessed with exosomes, like we talked about, the drug delivery superhighway of how our cells talk to each other, and my fascination. But there are things we can patent. We can patent the process. We can patent whatever we do end up making, that our compound, we can patent that, right? Like if it has specific things, and it will, like a fingerprint, those are specific things that we can patent and build a moat around the molecule. And this is what we have to do to attract investment dollars, right? So pay for the clinical trials, because phase 1/2A alone, we're talking $50 million or more. This is not a vacation to Mexico, right? So that's why I'm so excited to be involved, fortunately, at every layer of the process, and also get to be involved with people like yourself who have experience using these therapies to get people better, right? There's a very small number of examples, but Vertex Pharmaceuticals is one of them, an incredible company where the scientists directly interface with patients that have cystic fibrosis and get their approved drug for cystic fibrosis. And these people are suffering. These are teenagers to kids. And, the scientists pick up the phone and talk to the kids that are getting their drug that they develop. I mean, I don't know what's more gratifying, right, as someone in biotechnology than something like that. So when I look at my company and my team, I knew who I wanted to be like 10 years ago. I know who she is. Her name's Daphne Zohar. Her model, where Daphne and her company, PureTech Health, like she thinks like me, right? I should say I think like her, right? Because she's much further ahead than me. But at the time when I first ran into her, she's based in Boston and she's building a conglomerate. She's not building a company, she builds companies, right? So she has an umbrella, she has a technology incubator, a technology farm. She finds the right people, she finds the right technology, she brings in the experts, and that's really what it's all about, right? Because people build companies at the end of the day. So technology is wonderful, but a patent is just a piece of paper until a group of people pick up the piece of paper and build something out of it that can help people, right? So I like to think like this because this is how I was raised, and I it's not just because I set the bar higher for myself, it's because of the company that I've been fortunate to keep, people like you, where I feel responsible to push myself like this, right? And this like Elon Musk mentality, it's not just to go out there and like make as much money as possible, but it's about, okay, I'm a person that has a little bit more bandwidth than most from what I've gathered. My output capacity is a little above freakishly normal, right? And so I want to harness that energy for good. And, this has been my biggest problem as a builder is I always have a lot of ideas, right? And originally I was a great idea guy, but you know, I'm actually more proud now that our company is so focused on just 2 to 3 things, right? Because ideas are wonderful. Don't get me wrong. I'll sit here and talk ideas with scientists all day long. But until you can take an idea and do something with it and execute on it, which is a 10-year thing, right? And that's how I think now. And that's why I enjoy what I get to do so much, is because I'm not the scientist doing the thing. I'm not the Shawn Tierney injecting the soup and seeing the patient go,
Guiding
whoa.
the injection, yeah.
But I am the guy in the middle that will find Shawn and find the scientist and find the equipment and find the hardware and find the consumable and call the vendor when it's back ordered and call. I will do whatever it takes, right? So I think the journey, like we said earlier, is the best part. And it's cool to be a part of, like, going after a field in a space where there's no goalposts. It doesn't exist, right? And what's beautiful is, you know, since I started, there were no companies in human clinical trials. Now there's over 10, right? Now there's approvals almost. We're heading there, right? There's just over the last few weeks, Aruna Bio, the first neural exosome IND approved. That company has been working on that for over 7 years. These things don't happen overnight, right? And I just feel fortunate and blessed that going all the way back to, you know, 2008, 2009, and then transitioning into the commercial world and then coming on board with Chimera, you know, now all of a sudden our company has, you know, licensed one of the greatest exosome patent portfolios generated on the planet. How did that even happen? You know, it happened, right? And so now we're responsible for taking that scientist's 20 plus years of work, and it's not the hardest part, but it's just as hard as what he had to do, because we have to go make it practical, right? Science is not easy. I'm going to be the first to admit, but these are small batches on a bench in a controlled manner that are going into a mouse.
Yeah.
They have IRBs, they have regulatory oversight and scrutiny, but the safety profile and the uncomfortability bar is a little different, right? I'm telling you, producing something at scale in biotechnology,
It's no joke.
give it a
Yeah.
swing, man.
Yeah,
you know,
yeah,
and
I
you're
believe you.
you're gonna feel like a different person.
It's
So
no joke.
and it's just like the scale that you do, right? Which is why you're one of those diamonds in the rough, man. And from the moment that I met you, I was tractor beamed to you, because I was like, whoa, this guy, you know, I'm not saying you have it all figured out, but you have an open mind and you're willing to problem solve and think for yourself. And that combined with all of your expertise in pathophysiology and your decades of ultrasound guidance, and the fact that, like, the field was only using X-ray and fluoro for so long, but look what ultrasound can do. It offers this dynamic ability to see the nerve and, you know, see what's going on in real time. I mean, dude, this is incredible. This is amazing, right? So
Yeah, it's incredible to be able to help people in an innovative way. I think it's the innovation that makes things exciting. And I wanted to go through, hey, what can people get? What's available now? Now we have the use of exosomes for cosmetic use, and we have the use of exosomes maybe in other countries. But can you use, you know, you can now harvest your own cells and make exosomes from those cells and re-administer them back to yourself, as I understand, right?
yeah,
Right?
I
So
mean,
that's-
look, this is a great topic to be had, right? And I think the FDA's perspective on autologous biologics, where they come from us, come out and go back in, as well as allogeneic, where they come from off-the-shelf sources, is constantly evolving. It just is, right? Like, I myself have been to last year the Cell and Gene Therapy headline conference where Peter Marks spoke in Boston at Brigham, you know, so I've been to the events. I've tried to observe. I've tried to pay attention, and even I don't have all the answers, right? So what I do know is that there are different filtration technologies available, where, again, you know, the big guys in orthopedics and the regenerative world and some of the larger names out there, you know, you'll hear them kind of look at these newer options coming out and poo-poo them to a certain extent. That's for many reasons, right? It's new, you know, these individuals and these conglomerates and these groups are doing and have done amazing things for the field, right? But with that said, they have they're doing controlled trials. They're doing studies. They're doing things the way it should be done, right? They're monitoring and tracking. And, I put in this soup, I put in this cocktail of fat or bone marrow or blood. I want to know what happens, right? And that's what we all have to shift to. And I'm not saying that the off-the-shelves and the allogeneics haven't been doing that, because there has been some of that. But I think everyone kind of needs, the tide rises for all the ships, right? So I think that I'm hopeful that some of these bigger names can be more open-minded. And I think maybe some of the more risk-forward ways to approach some of these cell-free options is autologous approaches, right? Taking substances from our own bodies, using some of these sophisticated filtration technologies, and just offering a different soup of a something, right? Where we're removing the cells and we're putting in growth factors or some alternative, right?
And the rule kind of is from like tissue to like tissue is kind of where we're okay.
And we know we're already doing this. Think about alpha-2 macroglobulin, right? Like when I first met Gaetano Scuderi years ago and was fascinated by the science because it was so simple, he is taking blood. It's a lot like PRP, right? But he's taking the plasma and he's using a filter that traps certain sizes of molecules, right? You think A2M is the only thing in there? No. There's a soup of albumin and
that's one of the things that we wanted to talk about, because that's definitely available right now.
Mm-hmm.
And initially we thought it would be just for, to take the inflammation down. But what we're finding now is like, hey, this solves problems. Pretty amazing. But so basically that's just filtered blood, and there's, you know more about this than me, but there's one way to take your blood plasma and filter it and try to concentrate the A2M. And then there's the A2M that's scientifically made by Scuderi's company that we use in our facilities, where it's a lot more expensive. I mean, probably 4 times as expensive.
You mean the off-the-shelf recombinant, or?
No, I'm talking about the the A2M.
His filter.
A2M filter that he has is gonna, that's the original one that was available. So we never changed it because of this works. Why would we change?
Yep.
Even though we can get something that's 1/5 the cost.
Well, that's why.
But it doesn't seem to be as effective. So why do we want to take a chance with our patients? But
I think this
what do you
speaks
see
to
as the difference between the two?
This isn't just the difference between the two, but this speaks to a greater issue in the field overall, which is, look, to a certain extent, this is still cash pay. Some of the bigger regenerative societies have worked very hard to get employer-sponsored plans, which are some of the larger. When you have a big company, they can choose sometimes what they want their employees to have covered, and there's been some tremendous progress there with regenerative therapies and PRP and bone marrow now being covered by some of these employer-sponsored plans because of the evidence, because of the data that's there with orthobiologics, which is incredible, right? Like, that was not a thing when I first got started, and we didn't think it was going to be a thing. But I think the bigger issue that this speaks to is this is a business, right? And for the longest time, this was cash pay. And so who's behind the cash pay? Well, you have the manufacturers of the medical devices that have to get them cleared with the FDA via the 510K process. And then you have the distributors or the resellers or the layers of the sales representatives, right? And so while there is well-established data on what these systems can do and produced, there's many ways to spin that information, right? And so I think regenerative medicine specifically, because there has been problems with what is the patient actually getting, right? Unless there's transparency from the doctor and they're willing to have a registry and say, this is exactly what I'm making and doing and where I'm putting it. And that comes down to liability, documentation, informed consents, like all the layers of the clinic, which I have been there to help with and help doctors get started as well, right? So this is a massive problem, honestly, because you have a kit out there that is, here's what it technically is. It's called the hemoconcentrator, okay? This is a kit. You can find 19 versions of them out there. Becton Dickinson makes one. A subsidiary of Medtronic makes one. I could go through the whole list if you want, right? What does this do? This filter removes water. That's it. That's all it does, Okay. So you have all of these companies out there saying that they have this A2M product, which again, we take A2M, alpha 2 macroglobulin. It's a molecule. It exists naturally in our blood. It is an amazing molecule, right? And Gaetano Scuderi is an amazing person. This guy's a jiu-jitsu black belt. When I first met him, he's like you. He's like one of these people where you meet him and you're like, dude, like you're coming into my little pot of people, right? So, but again, it's a very large molecule that exists naturally in our blood, but this is not PRP. So what happens is when we spin blood, okay, and I'm not just talking about PRP, I'm talking about blood. When we spin blood at high rates in a centrifuge, first we have to put the same amount of blood opposite each other because of centrifugal forces, right? The physics of the equation. There has to be, let's say, 10 cc's of blood here, 10 cc's equidistant opposite each other in the machine. And ideally, you're putting at least 60 cc's if you're trying to make PRP so you get enough concentrated platelets, right? When we spin that blood in a centrifuge, typically at least at 300 G, a lot of times the protocols are thousands of G. It just, again, depends. Are you trying to separate the cells? What are you trying to separate out? So if you spin blood at the desired setting, let's say for PRP, you end up with platelet poor plasma at the top layer, okay? You'll see these fractioned layers. It looks actually really beautiful, right? You've seen it so many times. So you'll take out the container from the centrifuge that just spun very, very fast for several minutes. The container will show 3 distinct layers. The top layer is platelet poor plasma. Under that is the buffy coat, and then under that are the platelets. And so the platelet poor plasma contains hundreds of proteins and A2M, and mostly water that's in our blood. It contains fibrin, vitidrin, vitronectin, alpha 2 macroglobulin, albumin, and a long list of other proteins. Then the buffy coat has white blood cells, leukocytes, and a slew of other material. And then the platelets are on the bottom. Why? Because this is a density equation. The heavier things end up on the bottom, right? And so normally with PRP, we would take the platelet poor plasma off the top, and we would essentially concentrate the platelets back into a small amount of that platelet poor plasma, concentrating the platelets. So we end up with a high bolus, at least a 4 to 5 times concentration of those platelets than you would normally find in your blood. And now these ortho guys have gone as far as they test the blood before they put the PRP. They want to know how many billions of platelets they're putting back in because they've got it down like you. When you say 0.2 cc's into the femoral head, they know, a little bit more and I'm going to get an inflammatory response, a little bit less and and that's the beauty of this field, right? Is it does matter. It matters what's in there and it matters how much. And it matters how you get it there and how precise getting it there is, right? So going back to A2M, we don't just make PRP from the blood, right? We can also take A2M, this molecule, out of there. And so what does the hemoconcentrator do, this A2M that's flying out there? Here's why it's become so popular. The filter only costs a couple $100. It costs maybe $20 to make. The resellers are out there selling it anywhere for $100 to $300 at times. But again, what does it do? It takes the platelet poor plasma, attaches it to a filter, and only removes the water. What does that do? It concentrates the protein. You think about a solution of fluid. If you have Skittles in water and you take out the water, you have more concentrated Skittles, right? It's the same thing. So what Gaetano Scuderi actually invented was a patented, sophisticated tangential flow filtration filter, okay? And so his technology is the real A2M. His technology, when I was selling the kit, it's more expensive. It was like $1,000 back then. And that was, 10 years ago? It was still flying off the shelf because it works.
Oh, yeah, it works.
But I think the data shows it's 7 to 8 times more concentrated than this. I'm just taking the water out of the plasma, right? So,
yeah, and you don't have the inflammatory mediators in there to the same extent either. So you don't have this, like I said, I don't want to have things go sideways. And sometimes just concentrating the plasma is more inflammatory than not. So the A2M concentrated from the Scuderi kit never inflammatory that I've ever seen.
There's some really cool things that you can do with our blood, man. I'm telling you, there's a company I work with where they're the only cleared kit to have a hyaluronic acid additive in there. They have this like bio incubator, they call it, so they can take the platelet poor plasma, and they can essentially, it's a version of heat, but in the end, what it does is it creates like a biofiller from the patient's own body. So I don't know if you know much about aesthetics and medical injectables, but these fillers can turn complicated very quick. If they hit a vein, they get vascular occlusion. The complication rate in med spas is sky high. Why? Because they're not using ultrasound a lot of the times, even though thankfully that modality is growing in med spas.
Yeah, it is.
They're blindly injecting. They go to a course for a weekend, and all of a sudden they're an expert injecting the lips, injecting the tear troughs. This is intense, man. You know what I mean? So if you can make something more natural from the patient's own blood that is very similar to some of these products where you're paying med spas at times hundreds and hundreds of dollars for Juvederm, which is an HOA substitute, but you can get it in a PRP additive kit for $100, $150. There are other ways to go about these things to get just as, if not more effective, more natural versions of these products that will have less complications. They'll absorb naturally into the body. They're from the patient's body, right? So, I think this is the exciting part of the field, right? Is these big companies have such strong market share, but these little companies can come in and innovate with more natural products, with other options, more
And safer products.
cost-effective,
Yeah, exactly.
safer,
Exactly.
right? And so, look, people care about safety. Why? Because they care about risk. As the founder of a biotech, risk management is my number one job. It's my number one job. And I didn't even know this was a thing when I first got started, So I think that business owners, especially in healthcare, we're talking about humans. We're talking about making alterations to their bodies and their faces. They're going to feel better if they look better. And if you're making their innards feel better because you're fixing their nerves, you're helping them feel better on a whole nother level, right? And that's why I get so excited to just grow our friendship and develop our professional relationship, is because we just get to learn and understand more about how what I do helps what you do. And in the end, that scientist that's for 25 years developed this exosome patent portfolio, one of the greatest things in his life is probably to see it come alive, right? But that can't happen without you. That can't happen without me, right? So that's the exciting part of this, is we're just stewards. I just feel like a vehicle, to help all these people come together. And dude, I'm telling you, like, it is the most powerful thing in life to wake up every day and be a contributor to a group of people like that. I feel humble and honored just to get to be a contributor. That's it. It's one of the main things I live for, right? How do those people feel because of what I've helped to bring together? And I'm just one part of it, right? And you do the same thing in your clinical practice and when you teach. And it's not just the impact of teaching someone to be an expert with a needle. It's teaching them to be a better human,
And a better thinker, and all the way around. And I think that it's just, there's a real satisfaction with having a good team. And I think that's, you have to acknowledge that if you want to be happy, develop a good team, put good people around you. You're going to be like the people that you surround yourself with. One of the things that one of the organizations that we were associated with was asking for was, how do we make, astronauts more tolerant of radiation in space? And then there's some thoughts on that, and I know you have some insights that I don't have. So that's, I think, an exciting thing, but it's a crazy question to ask somebody as well, right? So you want to talk a little bit about that?
It is. Look, one of the most important people in our world, our society, Elon Musk, is actively interested in, like, what's the backup plan here, kind of thing, right? Which I know most people maybe don't think about that on a regular basis, but I assure you scientists do, and there's already been drug approvals with the FDA, Keytruda and drug derivatives where They've made discoveries in space because of microgravity or the elimination of gravity and the effect that that has on our cells, right? I don't know if you're familiar with the twins that were tracked and monitored. One went up as an astronaut, the other did not, and they saw incredible changes and differences in these two individuals, right? Not just in their epigenetics and how it changed their DNA and in their bio age, but in their emotional behavior, their every layer, right, of the person was different and changed in meaningful ways. So, look, I'm very fortunate. Again, this goes back to the people. I have a guy on my team. His name is James Seacrest. James, if he gives, like, I love when he's on calls. When he gets on a Zoom call and it's a first one and we're doing introductions, I'm like, James, you go first. You know why? Because he has just had one of these backgrounds where it's like, dude, how did you even do all these things, first of all? And like, because of the progression of the things that he's done, he started in the Navy, he was packing parachutes, he was doing engineering, he was doing a number of things in the Navy for 8 years. And then he became a chiropractor and got, very good with osteopathic manipulation. And then he was always interested in animals. So as he was paying for chiropractic school, he was a vet tech, and he was learning about the research aspect of working with animals and studies. And then, all of a sudden he realized, like, I want to get into research. So he gets a job with Charles River Laboratories, MPI Research, which happens to be one of the largest clinical research organizations in the world. And it's just a few minutes down the road from our company. So, he then transitioned to a Siemens molecular imaging. He then transitions to Astellas Pharmaceuticals. And then he transitioned to space research. And so James now sits on the Interplanetary Committee for, I'm going to get this wrong, but it's some wonderful board that meets, and it's an entity set up for space research. And so, look, James and I have had many discussions about how when you're in space, there's certain things that you can do to open up the blood-brain barrier. So, look, a potential cancer treatment of the brain, like a glioma or a glioblastoma, could involve sending someone up to space or simulating anti-gravity, microgravity here on the ground. And here's what's really cool. We've done this in the lab already. So James has a device that it rotates in a manner where it simulates microgravity, and we can put that in the incubator. And so we did this with the dental pulp stem cells, and it's really amazing what happens because, usually typically with cell culture, you need adherent substances. You need a flask, you need some sort of vessel where the cells will attach so they can multiply and they can grow, right? If they can't attach, they get confused, they feel wonky and wobbly, and eventually they die, right? However, when they're exposed to microgravity, they curl up into little balls because they think they're falling, because they are.
Right.
So they don't die.
Right.
But instead, they secrete really cool things. And so James has done extensive research, and there's tons of research now going on where they send these CubeSats, these little boxes where they create a lab on a chip in a teeny little box, and they shoot it up, in a satellite, and you can pay to have a little portion of the satellite to go up and run your experiment, or, you can work with NASA to do the same, and they'll take it up on the space station. And I think this is incredibly important for microbe research, bacteria, and, as we look at, potentially another planet or living on space stations. I don't know if you ever talked to someone that's been affiliated with NASA and a space station, but have you ever thought about like where the bacteria goes in there? It doesn't go anywhere. It stays there, right? So the biofilm thing up there, it's a massive problem, right?
Oh, yeah.
And so, look, there's already tons of research going on here, and Peter Diamandis has created his X Prize for space research. My very close friend and collaborator, who I'm going to see in just a couple of days, Dr. Ian White, he started a Space Aging Research Institute. So, look, there's some very powerful, very smart people that are doing these things. And I think, we're already seeing real-world applications with Keytruda and other drugs in development, and I think we're just getting started, just like with exosomes.
Yeah, and the other issue that with space, I understand, is how do we make our cells more tolerant to these cosmic rays, right? So that's another question that's coming at us. I think that if we're going to do this, because in our within the magnetic field of the Earth, you have a certain amount of protection, and once you leave that magnetic field, then you have an exposure to cosmic rays that's a radiation exposure. That's really the idea of going to Mars, is almost impossible without addressing that issue. And I think that it's hard to block. As I understand, I'm not an expert. It's hard to block those cosmic rays.
Yep.
So how do we overcome that?
This is a great
situation,
question, man.
So,
I took astronomy in college.
Yeah.
It was incredibly hard.
Yeah.
I took physics. I got a B+, right? So I don't know if I'm the right person to try to explain some of these things, but I do know these are fascinating topics. I know I do have the keen ability to find the people that know how to explain and discuss these things, and that's the coolest part about what I get to do.
So I guess what I'm saying is there's an effort to improve the resilience of human cells, right? And that's out there. The other thing that comes out there that I hear rarely about, but I do hear this argument is when we give somebody chimera, then there's no DNA in there. It's a microRNA from some cellular excretions for the most part.
Mm-hmm.
Is there a risk of changing somebody's genetic code? I think we're always changing our genetic code to a certain extent. There was some rumors that, hey, this is changing your ability to read your DNA, or you become a chimera. I think we're all chimeras to some extent. We're all picking up DNA from everybody we touch, everybody that's around. Dogs are great at it. Little kids are great at it. They're always like everything in the mouth,
Yeah.
And there's this idea that we pick up DNA, and our cells, our bodies pick up DNA, like from other people, and might just decide, hey, I like that DNA, I'm going to use that DNA, I'm going to incorporate that into myself on a grander scale from like somebody who had an organ transplant. All of a sudden, their DNA all looks like the person who gave them the organ transplant, and how does that happen? Now they don't have to take any drugs. That's fantastic. But just from a day-to-day experience, I think we as mammals or a race we're always evolving genetically, not only through sexual reproduction, but also from the DNA we're exposed to. Do you have any comment about that argument on the exosomes changing your genetic structure?
Yeah, of course. Look, I think that this is really, it comes down to a safety discussion, right? But I think in order to have an adequate safety discussion, you have to understand the science. And so, manipulating and editing and altering DNA, this isn't sci-fi anymore. This is already happening, right? Jennifer Doudna and, the other group that discovered CRISPR and RNA interference well before that, the ability to silence specific RNAs that halt specific production of specific proteins. This is nothing new, right? We just saw a few of the first dosings of the first gene therapies with great success, for some of the most rarest and horrible diseases. So I think this is just getting started, but I see the answer to this safety question with exosomes and, epigenetic changes as actually one of the more exciting parts about exosomes. If you look into and educate yourself more on some of the promise of exosomes, it really is the RNA. Right. And we've seen the explosion of RNA medicines. And I think when people think about DNA, especially the layman, what typically they think about first is the central dogma, right? It's called, which DNA makes RNA, transcription, RNA makes protein, translation. And this has been the school of thought for a very long time, since Watson and Crick, and, the X-ray crystallography work by Rosalind Franklin confirmed the structure of DNA, right? This is one of the original topics that got me so fascinated, and this was it, actually, right? And my co-founder, I walked into his office at UChicago like a year ago. He's got a picture on the wall with him and Watson. I was just like, is that Watson? So these things they're precious to us in the scientific community. And I think there's answers yet to come from the central dogma, but I think what's so important to realize is we've already shattered the thought process of the central dogma being the only way. That is long gone, right? And so my company is very focused on affecting the central dogma in reverse. We're actually trying to reverse engineer changes to your DNA via epigenetics, right? And so there's so much study on epigenetics now and increasing your telomeres and studying your age. And we've seen the work of David Sinclair, and we've seen the work of Altos Labs and all these companies getting billions of dollars of funding to study aging. But look, at the end of the day, there are precision approaches that can be taken now, that the FDA is already looking at, that companies are already producing and approaching them with, that involve post-transcriptional modification or post-translational modification. These processes, when you go and study them and the biochemical pathways, and I'm not just talking the organic chemistry as it relates to the pH of these activities and the glycolysis and Krebs cycle and the production of energy and glucose and pyruvate and their involvement in the basics of biochemistry, right? The things we learn first. Let's take a step back to just the machinery of the cell. This teeny little cell has such sophisticated machinery and capabilities, and a lot of this is related to DNA, RNA, and proteins, right? The ribosomes impact protein production, the endoplasmic reticulum. Every single organelle is perfectly placed in a beautiful symphony to interact in some way, shape, or form to affect DNA, RNA, protein, right? Why do we study? Why do we eat protein? Why are we so obsessive over protein? Amino acids, the building blocks of protein, this is being human, right? So when we look at leveraging exosomes, when we look at leveraging exosomes as delivery vehicles for gene therapies, which is happening, Sturm Bio, and I could list 7 other companies, right, that are doing this. There's many ways to approach the exosome. It could be a delivery mechanism. It could be a targeting mechanism. It could be a biologic soup that we're introducing, right? So we're very interested in microRNAs, specifically, and small interfering RNAs. There's been some work done on RNA interference. A lot of funding went in there in the 90s. It was an area that I read tremendously on and tracked and followed extensively, but not a lot came from it. Actually, lately, there's been some activity kind of bubbling back up. But I really, the audience is probably not aware, and, you're probably not aware. There's 7 subspecies of RNA, okay? So there's long non-coding RNA. There's rRNA. The list keeps going, right? Yeah. And much of this is non-coding RNA, okay? And so we have billions and billions of base pairs in our DNA, and when we have DNA is transcribed into RNA, it actually produces what's called introns and exons, okay? So when that transcription process occurs, there's a massive strand, but there's intron segments and there's exon segments. Well, guess what? Part of the intron segments cut out, separated from the exons. One is only brought together to actually translate into a protein. So we have really all of this nucleic acid information that's sleepy. It's dormant, right? And if you look at the structure of DNA, it's actually wound up very, very tightly in what are called histone proteins. And these enzymes called histone deacetylases and the process of methylation can slowly open up, can slowly change, can slowly alter the structure of DNA. So that's what's so beautiful about biochemistry. And that's why I love the science of the cell so much. I had a professor that he taught me all these things, and it just got me so obsessed in understanding there are so many intricate mechanisms and just understanding that central dogma. And now we have the ability to potentially reverse and go the other way with the central dogma.
Yeah. So you can take you're basically taking RNA, and your body may decide, do you like what that RNA is transcribing or what that RNA is doing? And you may actually design some DNA in your own cells based on that.
Or you're manipulating RNA in a way where you're just changing the expression of specific DNA. You're keeping this
gene
quiet,
expression,
you're turning
right?
this on, right? Gotcha. So that's the process of methylation, which a methyl group is a carbon with 3 hydrogens on it. This is also the beauty of the chemistry, right? there's only a couple of molecules that are involved in this, carbon and hydrogen, in this process. So I think as scientists, we understand the powerful nature of DNA, RNA, protein, but especially now more than ever, when we look at the process of translation, which again, when we talked about all that sequence coming together and cutting out the riffraff non-coding RNA sequences, and then in order for it to be translated into a protein, that strand actually folds over on itself. It's really beautiful. You can watch this on a YouTube video. But all these little proteins called transcription factors, they have to line up in like just the exact right way. And there's this cellular machinery that brings them all together. And then when they all come together at the right moment, it splices it and creates a protein with the ribosome, right? So I get a little too weirdly excited about these things because I'm very interested in them. But, these weird words have meanings. And I think being able to attack the meaning of these weird words and these cellular structures from a different way, from a different angle, in a different direction, it opens up very exciting possibilities for therapeutics. And this is already happening. Just the other day, the first IND approved, the first human studies approved to start on a targeted sequence-specific RNA for tumors, where this therapy is so selective that it only targets the cancer cells. It only kills the cancer cells. That's how specific this company engineered this lipid nanoparticle to deliver these
now
RNAs.
We're talking about these lipid nanoparticles, which, when obviously they can kill cancer, and then the interesting or the famous, application, the most controversial application would probably be the COVID vaccines, right?
Of course.
So, how does that differ, from what we're talking about? So as far as these cellular excretions.
This is a great question. And again, this comes down to the drug development process and the ability to control that process, right? So you may have heard of liposomes before. There's a lot of supplements like Chris Shade's brand of Quicksilver and some other good brands that use this liposomal delivery system, right? Because we know we take a vitamin, if it's a regular pill, the microvilli in our intestines, they're probably not going to absorb a lot of that. We're just going to end up urinating it out. And so they've come up with these liposomal technologies, which is essentially, again, a little bubble of fat, a lot like a cell, but even smaller, without the nucleus, it's just synthetic, little bubble of fat, much like an exosome, which is non-synthetic, to deliver things, right? So, this is an interesting area because lipid nanoparticles like liposomes are synthetic. If you go to venture capital meetings in biotech, and you witness 20 companies trying to raise money right now, 19 are lipid nanoparticle platform companies, right?
So it's the future of
it is the future,
yeah.
But I think it's also too important. For me, this is very important professionally because it's my job to help the field understand that an exosome, in my mind, is actually a far superior version to a lipid nanoparticle, right?
Right.
So
there's
and why, right? Well, it's not synthetic, right? This is native. This already exists. We talked before how cells talk to each other. It already has the proteins on the surface, the ligands, the interactions to have a natural interaction with your cell. There's already an internal process to absorb or phagocytize that exosome. And then there's internal cellular machinery where it interacts with the lysosome, which is like the garbage man. It gets reprocessed, different things happen, and then more are produced, and it's exocytized. So they send them back out, right, to go do other things. And so that natural process and that natural emphasis, to me, it just makes more sense. And lipid nanoparticles and the vaccine itself, I think, again, without us going too far down the rabbit hole, even though there's just more and more evidence that there wasn't enough testing and there wasn't enough, understanding of where these, like what we talked about, the PK/PD, where are these going? What are they doing? What are the toxic effects? And I think as we see more and more people come down with vascular conditions, and heart conditions, which this is not conspiracy theory, right? There's been all these issues with myocardial problems and in young men especially, right? This is very scary, and clotting and all these vascular related issues. I think exosomes potentially are a formidable answer. And our inventor collaborator has already shown this in animals. So
yeah, I think we've seen that happen as well. Yeah, so these are not the same as the vaccine. So just want to clear that. Not the same stuff. It's a little bit more natural, a little bit, a little bit much safer, and thousands and thousands, hundreds of of people have had safe experiences here. So the other big issue with exosomes, I would say, is that this new field, or this idea of, we're not just going to do mesenchymal exosomes, but there's endothelial exosomes are like the new buzz. And I think this is probably one of the most exciting things for me recently. I mean, we've had, because these endothelial exosomes look like they're really showing some changes for vascular problems, for cardiovascular disease, for plaquing in the arteries. But also you have to think that, our organs are lined with endothelium. This endothelium is kind of the inner skin. A lot of inflammation is happening and derived there. So these exosomes could potentially be even more groundbreaking. And I'm just speaking from my understanding. You might tell me I'm wrong. But also there's a potential that we can just take our own blood, get these endothelial progenitor cells, and expand our own exosomes and give them back to ourselves, and have a very safe experience that may even be okay to do in this country.
Yeah, I think this is, again, like you said, one of the more exciting areas of what's coming. I think we've already kind of discussed this progression, right? Like, this is the Gen 2, the Gen 3, the Gen 4. And this goes back to our original discussion around Dr. Arnie Kaplan, the late, the great, and, how he changed the name from mesenchymal stem cell to medicinal signaling cell, and how we really need to start pivoting the discussion away from, I'm frankly, man, like, I'm tired of seeing all these companies out there that are commercializing MSC products. They have very little understanding of what these products are and what they do, right? And I'm not being negative. I'm just saying where we are headed is something much more specific, right? Because what these cells actually are is they're parasites. They're the resident satellite cells, the resident native cells that are the most juvenile, you could call it, for that tissue, for that region, right? You go to the liver, like we talked about, you're going to find stem cells that carry a liver-like phenotype. You go to the kidney, you're going to find stem cells, parasites, the satellite cells. They're looking, they're monitoring, they're sensing what's going on in the environment. These are not static cells. They're dynamic. They're looking around, they're sensing what the environment needs, and then they're secreting those factors, those trophic factors, those growth factors, those cytokines inside of exosomes, via the paracrine system, it's called, into that native environment, which hopefully is maintaining homeostasis, right? And you made a great point about DNA earlier, and I got super excited about all these cellular mechanisms. Well, guess what? There's tons of cellular mechanisms that already exist to find foreign DNA and kill it, right? We have tumors in our bodies all the time, but we have native endogenous archaic mechanisms that already exist to find foreign DNA, kill it immediately, right? So, look, I think that this has already been shown in animals, in large animals, in swine, in rhesus monkeys. This is one of the most promising next areas. This is an area where we're heavily vested and have an extensive patent portfolio. And so, again, I think now that we understand what an MSC actually is, right, which is a native progenitor, juvenile, nearly less undifferentiated cell type for that region, where the cells grow off of and stimulate off of that cell, all the other cells in that environment, and it dictates the health, the wellness, the vibe, and the feeling of that environment, right? Like, what's the radio station that's playing in that tissue niche? Now we can really start having the real conversation. So there's a paper by Melvin Hayden. Unbelievable. This guy has an incredible ability. Johns Hopkins, all these, Harvard, these major institutions, they have amazing people. But then all of a sudden you find a guy at the University of Missouri, like, he's actually the guy. and he's just under the radar, just doing his thing. World leader in electron microscopy. And he's taken pictures of this. He's shown in models of obesity and models of diabetes that these resident parasites, these resident MSCs, he's shown how they behave and how the vascular spaces are impacted by inflammation with imaging, with seeing it, right? So this is not like theory anymore. And this publication that he put out, I'll send it to you, but amazing imagery throughout the whole publication, right? And so seeing is believing, especially with exosomes, right? So using visualization methods like atomic force microscopy, electron microscopy, cryo electron microscopy. We have multiple collaborations with the University of Chicago imaging cores and some laboratories that have this expertise. And so, again, you're not just how many particles do we have? This is not a how many particles do we have equation. If one more person says how many exosomes, we're going to punch them in the face, right? Because there's so many other things that matter.
Yeah,
because remember we talked about the morphology matters too. Remember DNA? DNA is just not DNA. It's how it's organized and the morphology and the shape of the DNA that dictates its function. Right? And we know this in chemistry, chiral structures and all the things that enzymes and the lock and key fit model. And so, again, I think that there is a growing number of the population that are affected by these vascular diseases, vaccine or not, COVID or not. I do think that there's more and more literature and studies now coming out that even if you've just had COVID, which you were talking to me about before, the cardiac implications are tremendous.
Oh yeah,
tremendous.
That's my kind of speech that I have to athletes, because the athletes are in some way the vulnerable, untalked about. Because an athlete, a fit athlete, a lot of times will get COVID and won't have a cold, won't have a flu, won't test positive for COVID, won't have any symptoms at all, except for all of a sudden they have some cardiovascular problems.
Cardiac output, right?
Yeah.
Changes.
Well, cardiac output, vascular changes, heart rate variability, but more than that, clotting and vasospasms and heart attacks and strokes in young, healthy, world champion, elite athletes. And we've seen that too often. And it seems like it happens sometimes, I think, after somebody, for a non-athlete lessons, people will get sick and they'll have COVID, maybe some mild symptoms, and that's not that severe for most people. But like a few days after they're sick, then maybe that starts to affect, just my observation. But it seems like it's starting to affect the cardiovascular system, and then that's where people are vulnerable. So I think every step we could take to avoid that becoming an issue is it needs to be taken.
Yeah.
And the fact that we have a technology that may be available to people in the United States, because once we actually harvest this, my understanding is that we could probably bank that and then redistribute it to somebody when they need it. So
yeah.
Okay.
And this is not just ideas anymore at this point, right? I mean, we have animal work showing infarct reversals, thrombotic reversals, right? And so, again, we go back to our basic physiology class. What's the first thing we learn about? We look at all the different tissue types under a microscope: cuboidal tissue, endothelial tissue. This is the first thing we learn. We have to learn what it looks like. We have to identify the differences, right? And one of the most prominent tissues throughout our vascular system is endothelial cells, right? And so these cells are pivotally important in the activity of blood flow, which we all know how important nutrient delivery and good health is for vascularity. And what are the two main emphasis of stem cell therapy? The introduction of growth factors, right, which means a reduction of inflammation. People are inflamed, so we're going to reduce their inflammation. And angiogenesis, the incitement of angiogenesis. So we increase vascularity to that issue. We cannot heal disease without bringing blood flow and nutrient flow to the tissue. If we have blockages, if we have thrombotic cascades, those cascades go in a specific direction for a reason, right? And so we have some studies, these are incredible studies, right? Where we grow Schwann cells, we grow endothelial cells. At times we co-culture them, and then we compare the profiles of what they secrete. It's called the proteomic profiles. This is thousands of proteins. They're very similar. but they're not completely similar, right? There are nuanced differences in those proteomic profiles, and introducing those endothelial cell-derived small extracellular vesicles, we call them, it has incredible potential for reversing a lot of these vascular issues. And this is pivotally important because, you look at the work that's been done in stem cells for cardiac diseases, and there hasn't been tons of progress, right? Because there's not a big regenerative impetus to cardiac stem cells, right? Like this is not a thing. So if we have a way to improve blood pressure, improve vascularity, I mean, diabetes, these are the biggest issues afflicting our country and our people. Right?
It's huge, and it's scary huge, but I think it's extraordinarily promising. So I think I can't say enough. That's endothelium. From just the orthopedic perspective, now that is also the lining of our joints, right? So our joint inflammation is also associated to this endothelial dysfunction. And this stuff is looking promising not only for vessels, but also for joints and also for nerves and neurodegenerative problems. So it's a big deal. It's a big deal. Such a big deal that it's scary, and I think it's scary to a lot of people. But I think it's something we have to look at, something we can deal with, and I'm looking forward to ways we can do that. Maybe some ways in this country, maybe some ways in other countries. We're trying to figure that out and make it happen.
I think without a doubt, offshore, we'll be testing some of these things on athletes in our community, and individuals who have, again, they seek out treatment and options like this at a higher level, right, and oftentimes can't find it domestically. But I do think there's some low-hanging fruit with a risk profile that's acceptable here in the U.S. to just do some of these things with the patient's own cells, right, with a very low risk and a very low bar for issues.
Certainly, if it becomes available to everybody in my family, whenever it becomes safe and available to do. If somebody has COVID or a cold or a flu, I would like to have that cell banked, and I'll say, hey, send me a few vials, because I just don't want this person to have a heart attack or a stroke, because they're important to me, and I'd like to see this life go on a little bit longer in this way, right? So it's super exciting stuff. Is there anything you want to add? I just super appreciate if you being here. I think we've covered a lot of bases.
I think I really valued this discussion. I think that we talked about much more than stem cells and exosomes. We talked about being a good person. And that's the people that we like to attract and spend our time with are those doers and the people in these circles that they just want to move this forward, man.
Right.
They want to help people, right? And so, there are the layers of the reps and the distributors and the conferences, and these are needed layers. They're not the big bad wolf either, right? So I think more dialogue, more discussion, and pushing people like yourself up to the top, on these podiums and with the ultrasound trainings, and people need to be learning from the right people, right?
Yeah.
I think that's one of the biggest problems is a lot of times, a lot of people think they're gurus in this space, right? Because they did a few injections. So I think that, barring being negative at all, we just need to take a couple steps back as a field, as a domain, and make sure that the right people are the teachers. And also, the clinicians and the builders and the regulatory bodies need to talk more, right? Because the FDA has a lot of those resources, like we touched on, right? They do exist. And so I think being factual and presenting what's real and what's true, right? Which is at times difficult to do in our society nowadays, right? Like we talked about, with common media and what's available and out there, it can be hard to know, what's real and true. So, you're someone that thinks for himself. I don't think you're easily swayed or easily influenced, right? And I think that to a certain extent in our world, thinking for yourself has maybe waned a bit over time.
Yeah.
So we'll have to do this again and maybe talk more about that.
I like that. I think that I always think that it's hard to get closer. I mean, it's hard to get closer to reality. It's hard to get closer to the truth. And our minds easily sway the direction that we want to go. So, it's kind of a challenging thing for a professional of any type. I think that you and I share this, is that you have to go into a situation thinking, everything that I know may be completely wrong. And if you go into that situation and you say, I don't know what the answer is, but I'm pretty good at figuring things out,
And
then,
I'm not going to quit.
And I'm not going to quit. Then you can get some real answers done, and you can get closer to the truth. And that's like just as close as we get to the truth, for that patient, for us, it's just when we're doing that consistently, then we can start to ask more of our doctors, of our clinicians. And what I mean by that is, I think that doctors and clinicians have become okay with people not getting better, with not solving problems, because it's so hard, and in our system, it's kind of, in many ways, it's not possible to do things the way that the standard medical system is set up, with insurance and all the regulations and the oversight that happens there. But I think we need to start looking at things like, hey, let's look a little bit deeper, and let's solve that 7th, that 8th, that 9th, that 10th problem, and look. So we see a more complete aspect of this, a more complete. We're talking about very in-depth ways of looking at, cellular biology and treatments. So I think we have the technology where we can solve a lot more problems, and doctors need to start to expect themselves to be able to solve problems, and then we can move forward in a good way. So I hope that's what we bring to the table.
100%.
All right. Very good. Thank you very much.
Thanks so much.
Much appreciated.
Yeah. Cheers.
Yeah.